Hi, everyone. Welcome to today's seminar in our seminar series called Cutting Edge Advances Toward Personalized Mental Health, hosted by the Center for Precision Mental Health. I'm Xue Zhang, and I lead this seminar series along with my colleagues. I will begin with a few housekeeping items. So this seminar is recorded and will be posted on our website next week. You can scan the QR code shown on the left side of the screen or use the link in the Zoom chat to visit our webpage. From there, you can also sign up to receive announcements about future seminars and events. If you're interested in joining our center as a research member, please scan the QR code on the right side of the screen or use the link in the chat to complete a short form, and we will follow up with you shortly. Today's seminar is a hybrid event. We will take questions at the end of the presentation. For those attending in person, our staff will bring a microphone to you. So please raise your hand if you have a question. For online attendees, please submit your questions using the Q&A box, and I will read them to our speaker. We will alternate between the questions from the in-person and online attendees. Now, it is my pleasure to hand over to the director of our center, Dr. Williams, for the introduction of our special keynote speaker.
Thank you, Shui. Welcome, everyone, to this Cutting Edge Advances Towards Personalized Mental Health seminar series. We're thrilled you can join us both in person and online. I'm Leanne Williams, Director of the Center for Precision Mental Health, and it's truly a pleasure to have you here today. It's a great privilege to introduce our keynote speaker, Dr. Weidong Cai. As some of you may know, Dr. Cai is a clinical associate professor in the Department of Psychiatry and Behavioral Sciences here at Stanford University School of Medicine. He earned his PhD in psychology from State University of New York at Stony Brook, and there he focused on neural mechanisms of inhibitory control. He then completed his postdoctoral training at UC San Diego using functional magnetic neuroimaging and transcranial magnetic stimulation, TMS, to further probe inhibitory control in the human brain. Since joining Stanford, as you'll hear, Dr. Cai has built an innovative and highly impactful research program. It's centered around the neurocognitive mechanisms underlying ADHD and related disorders. I've had the privilege of working with Dr. Cai during his time here, and it's been deeply rewarding to watch his program grow into such a strong and distinctive body of work. His research is supported by multiple federal and foundation grants. They include NIH, Mentored Research Scientist Development Award, and you'll see he takes an interdisciplinary approach where he brings together cognitive science, advanced fMRI, and computational modeling. As you'll hear, his work examines cognitive control systems across the lifespan and how their disruption contributes not only to developmental conditions like ADHD, but also to disorders later in the lifespan, neurodegenerative disorders. And he's very committed to translating these tools and these findings into more precise treatment strategies. So today we'll hear his talk titled The Maturation of Dynamic Dual Control Mechanisms, Neurodevelopmental Trajectories and Their Disruption in ADHD. So please join me in warmly welcoming Dr. Weidong Cai.
Hey everyone, thanks Leanne for a kind introduction, and thanks Xue for organizing the event. I'm really glad to be here to talk about the research from my team we conducted in the past few years. So today the topic is the maturation of dynamic dual control mechanisms. So here dual control, I mean the cognitive control system. So cognitive control, which is a very broadly defined term, we use it refers to our ability to coordinate multiple, often conflicting motor cognitive processes in order to achieve short-term and long-term goals. So classical frameworks such as those from Miyake 2000, Diamond 2013 studies, they break down control into three main components. Inhibitory control, the ability to inhibit inappropriate responses, working memory, holding information in online systems, and cognitive flexibility, your ability to flexibly switch the systems to different tasks. Today, I'm going to focus on the inhibitory control systems. So inhibitory control is important in our everyday activities. Take driving as an example. When you're driving passing the school zones, even if the street looks empty, you will always be very cautious and ease off the gas pass, right? Just in case some kids will run into a street. This is a proactive control of your motor functions. It's also about how to control how to resist temptations. Imagine your favorite dessert is placed in front of you. You have a long-term goal, you keep it healthy, and maybe also control the weights. You need to balance the temptation of delicious food versus your long-term goals. So this is also cognitive control. So cognitive control sometimes can be triggered by, okay. Cognitive control can be also triggered by unexpected sudden event. For instance, when you encounter wildlife animals outside, you need to determine what to do. Actually, this is a photo I took 2 weeks ago when I take a walk outside of my lab and run into a coyote. I was at the beginning very relaxed and take a photo of him until he started following on me. So I changed my plan. Instead of keep walking for 2 miles, I returned to a lab. So cognitive control is implied in many aspects of our life. So to understand cognitive control, we want to know how our brain functions and how our brain system develop from childhood to adulthood. So deficits in cognitive control is widely implicated in different kinds of disorders. Here's a literature search I did last week on PubMed. You can see cognitive control deficit impacted in a variety of disorders from ADHD, schizophrenia, depression, bipolar to substance abuse. So understanding cognitive control deficit and its underlying mechanism, so which can serve as transdiagnostic markers to help us understand the broad landscape of psychopathology. So how do people study cognitive control and inhibitory control? There are many different paradigms like flanker task and Stroop task. People have been widely used to study cognitive control and inhibitory control. But in recent years, one of the most commonly used paradigm is a stop signal task. I will tell you why. So this is a classical paradigm people use to study monkey behavior. So you can look at this task. There's two kinds of trials. One is the go trial. Go trial is a trial without stop signal, and a stop trial is with stop signal. So monkey sit on a chair, and the monkey need to fix the eyes in a center fixation here, right? Then there's a center fixation disappeared. A target appears in the left or right side of the screen, and the monkey need to make a psychotic eye movement to shift the eyes to the left or right, depending on where the goal target it is. Then occasionally, in a small proportion of trials, like 20% trials, the fixation in the center will reappear here. So this is a stop signal. So it's shortly after the go signal occurs. So when monkey realizes there's a stop signal, they need to stop making psychotic eye movement. They need to continue remain the eyes fixed in the center without move eyes. So it's called the stop trials. So if they successfully stop their eye movement, we call successful stop trials, otherwise they failed. The nice thing about this paradigm is that Logan and Cohen in 1984, they developed a risk model. So risk model allows us to estimate a parameter we call stop signal reaction time. So how fast the monkey can cancel the response when they say a stop signal. This is important because usually we cannot measure how fast the person can stop response because there's no recording. You don't have the actual observation of that. But with this risk model, which based on the distribution of RT and the accuracy of the stop trials, as well as when stop signal occurs, we can calculate the SSRT, which is a critical parameter people have not been used to quantify the cognitive control and inhibitor control abilities. So how do we know this measure is accurate? Because it's a model-based estimation. We don't have a sense about whether it's accurate or not. So in a follow-up animal studies, people have recorded the neuronal activity in the frontal eye field. Frontal eye field is a cortical brain regions that's controlling the eye movement, either static eye movement and fixation. So there are different types of neurons. One is called movement neurons. Movement neurons shows increased firing rate when a monkey makes static eye movement. Another is the fixation neurons. So fixation neurons has increased the neuron firing rate when they are fixed in the center, do not move the eyes. So what they show here is there are very nice divergence between neural activity and deactivation. And the divergent point is kind of closely aligned to the SSRT estimate from Reed's model. So this gives confidence about the estimation of model. Of course, there's a lot of assumption of the model, and you need to make sure your behavior data meets those kind of assumption before you can apply the model on data. But people have used the stop-sign task to study the developmental of cognitive control or inhibitor control. And this two study shows the SSRT, how fast one person can inhibit the response. The speed can become faster and faster from early childhood, continuously improve through early adulthood. And we have reviewed those studies and identified the later childhood is critical period where it shows the largest improvement in SSRT, which happens to overlap the period clear scale diagnosis for ADHD. So we think this is a very critical period for us to understand how brain develops. So we're talking about the cognitive control and the inhibitor control, but those processes are not a unitary process. There's different components. So there's a dual control model proposed by Adam Aron and Todd Braver. Previously, they suggested the cognitive control systems operates in two distinct modes. One is reactive mode, one is proactive mode. Reactive mode is refers to how you be able to inhibit the responses when you detect a conflict. Like if you're driving through a school zone, suddenly you see the kids running across the street. You need to immediately press the brake, right? Stop, do not press the gas again. There are also proactive control mechanism, which means you have some knowledge about the forthcoming interference or conflict. You need to apply a proactive strategy in advance before any conflict has been detected. It's just like when you're driving through the school zone, even if there's no kids around, you'll be very careful. You need to release the gas a little bit, apply a proactive prevention strategy to avoid anything happens. So how do we study proactive and reactive control? So basically, how do we decompose those kind of process from the cognitive paradigms? There are a few different experiment paradigms we can use. So here I gave an example we used in our previous study. So this we call is a modified or conditional stop-sign task. So basically, it's a stop-sign task at some additional conditions. So in this task, there are three types of trials: the certain goal trials, uncertain goal trials, and stop trials. So if you look at these two trials together, uncertain goal trials and stop trials, they're basically a stop-sign task. You see a go signal, point to left or right, make a response button, and whenever that stop signal occurs, you stop your response, right? So on top of that, you have a certain goal trial. How do you differentiate a certain goal trial and certain goal trials? It's from the beginning as a queue. If it's a blue cross, which means there's certainly no stop signals. You don't need to worry about stop signals. They always go signals. In this case, imagine how something we do. They will always respond very quick, right? So if we try to define the motor cognitive process for every kind of these trials, we can imagine in the certain goal trials, there's only motor responses. In uncertain goal trials, there's a proactive control and the motor responses. Whereas in stop trials, there's a proactive, reactive processes. So based on these three different trials, we can identify or isolate specific reactive product components. For example, reactive control, we can use the contrast between stop and go trials, and proactive control, we can use the contrast between uncertain and certain go trials. So in some studies, I explain later, so we will also find the commonality between the go trials and the stop trials, because in go trial and stop trials, they both has proactive control components. We can look into the common activity pattern as a definition of proactive control involved in both trials. We also know that proactive control is not a static process, right? So every time the subject look into go signals, they are not always apply the exactly same amount of proactive control on the same trial, on different trials. So people always adjust their response strategy based on the trial history, based on their performance feedbacks. For instance, if you made a mistake in last trial, most likely in next trial, you will slow down, you'll be more cautious. And we have also used some computation model developed by Dr. Angela Yu who was UCSD. And they can use the computation model to estimate the trial by trial subjective belief of stop signal. So they can estimate the subject trial by trial belief, how likely the next trial will have a stop trial. So imagine if you think the next trial will likely have a stop trial, you will slow down. Otherwise, you will speed up, right? So we have used the model to demonstrate, based on the estimation of the subjective belief of stop signal, we have shown a nice correlation between the reaction time and the probability of the subjective belief of stop trials. So that's confirm our prediction that when subject expect the next trial is more likely to have stop signals, they will slow down. They will apply more proactive control strategies in the trials. So what brain systems involved in cognitive control and reactive control? So we can look into a literature. So here is the meta-analysis we did previously. The panel A is the meta-analysis from adult studies. So here are including both go-no-go and the stop signal task. Both are used to study inhibitor control. And there's the 7-day studies of that. We can see the typical salience network, including anterior insular, inferior front gyrus, and the pre-SMA, and also frontal parietal networks are activated commonly across different studies, suggest the systems are consistently engaged whenever there's a need of reactive control. We also did a analysis on kids. So the threshold is weak because there are few studies. Here is only 14 studies from children. But overall, the pattern is very similar. We see the salience and the frontal parietal systems are engaged during cognitive control. Previous study has also looked into the proactive versus reactive control systems in adults. And here we use our conditional stop signal task, decompose the reactive and proactive control, and look into the brain systems engaged in these two control systems. Although the proactive control shows the relatively weaker activation, but the key regions are involved, such as anterior insular and inferior front gyrus, as well as pre-SMA, which are salient systems engaged in both reactive and proactive control. So this is important because we have some knowledge about the similar system between proactive and reactive control engaged in brain functions. So we can create a template based on one control systems and see how likely another control systems engage the systems in the trial-by-trial fashions. I'll explain its details in the analysis later. So those are the conventional fMRI study, which tells us static activation during task conditions. But we are also interested in dynamic process because the human cognition relies on the dynamic and flexible, stable processes. One is a stable and flexible process got disrupted. It's often affected behavior, may lead to some clinical symptoms. So we want to understand whether the brain function can remain stable during task performance in a stable and flexible way. So today I'm going to talk about 3 ongoing and recently finished study from my lab. So in the first question, I'm going to ask, how does the brain system develop to support dynamic, proactive, and reactive control? The second one, I'm going to show you some of our published work to show like children with ADHD and demonstrate the deficit in reactive control systems. The last, I'm going to talk about one of our studies. We're looking into stimulant medication and how this medication affected the brain dynamics in children with ADHD. So in the first, we're interested in the neurodevelopmental of the neural control systems. So in this study, which is ongoing, we leverage two large scales neuroimaging dataset. One is the ABCD study. I think many of you are familiar with the study. It's a multi-site, including 21 site neuroimaging dataset collected in the United States. And this longitudinal study is recruit the kids from 9 years old and collect data every year. So we are using release 4.0. So we only have the first 2 time points from 9 to 10 years old and from 11 to 12 years old. We also want to understand how the developmental goes to the adolescence. So because we don't have data from ABCD yet, we use the data from imaging. So imaging data is collected in Europe, so which is starting from 14 years old and to the 18 and 22. So imaging data collection has been finished. So we kind of put these two data sets together to understand the neurodevelopmental trajectory from the late childhood to early adulthood. So in the ABCD study, we have over 11,000 subjects to start with, but after all kinds of like screening and hand motion and data completion, and also we want to have 2 sessions of the stop thinking task. So we only have about 2,000 subjects left. And for imaging, we have about 1,200 subjects left. The nice thing about these 2 data sets is both have a stop thinking task, which allow us to look into cognitive control. So first, I want to show you how the inhibitory control system develop from the late childhood to early adulthood. So this is a figure to show how the behavior changes through development. The x-axis shows your developmental stage. In the ABCD, 10 years old, 12 years old, imaging data set from 14, 18, 22 years old. On the left side is SSRT. Remember, SSRT is a measure how fast the person can stop the response, basically a measure for the cognitive control. On the right side, we use the IRV, is the intra-individual response variability, basically a measure like how stable the performance is. This is a measure actually a standard deviation of GO-RT. So the smaller value means the performance is more stable. The higher value means the performance is more variable. And so in both scales, SSRT and IRV, the smaller value means better performance. The larger value means worse performance. As you can see, from later childhood to early adolescence, we see a significant drop in SSRT and also IRV. And from 14 to 18, 22 years old, surprisingly, we didn't see continuous decrease of SSRT. So it seems to get into a stable period. In contrast, we still see the continuous decrease in IRV, so behavior become more stable, right? So when I see this behavioral patterns, the first thing coming to my mind is, does there like a brain system becomes more stable? So if the response variability become more stable, does their brain activation from trial to trial become more stable? So can we understand that? Can we study that? So we used the single trial model and the representational similarity analysis to look into this question. So this is led by my colleague, Dr. Zhiyao Gao. So let me explain to you the difference between the single trial model versus the conventional GRM models. Conventional GRM models, as many of you know, so we define the vectors to model the brain activation associated with task conditions, right? So for example, we have this stop thinking task. We have go trials, we have stop trials. So basically we can define 2 vectors if you want, define 2 vectors. One is the go vector, one is the stop vector. So go vector will model all the activation in the go trials. The stop vector will model all the activation in the stop trials. You get the average activation pattern for go trial and stop trial conditions. Whereas in the single trial models, we build different models for different trials. I give an example. For example, here is a model for the first Go trials. You will have one specific vector to model the very first Go trials, okay? Then you have a separate vector to model the rest of Go trials. There's all the other Go trials. Then you have a vector model all the stop trials. So this is the model of the first Go trials. You can do this for second Go trials, for third Go trials, and going forth, keep going on. So imagine if you have 100 Go trials, you will have 100 single trial models. Every model gives you a representation of that Go trials. So once you have those single trial models, now you can do all kinds of things to look into the trial-to-trial neural stabilities. For instance, whether the amplitude are different from trial to trial, and whether the spatial pattern are different from trial to trial. So you can basically develop some kind of metric, like we call the spatial similarity. For any pair of Go trials, you can ask whether this pair are similar or not. Now go back to our previous slides. So we see the behavior patterns becomes more stable. So now we can ask whether neural activation pattern becomes more stable from late childhood to the young adulthood. And this is what we found. So the y-axis is basically the pattern similarity. So there's a pairwise brain activation similarity between any pair of same kind of trials, Go trials or stop trials. And from the late childhood to early adolescence, we can see the increased pattern similarity. And from middle adolescence to the early young adulthood, we also see an increased neural stability. So this suggests the neural system becomes more stable around age. Okay. So next, we want to get to know about like the dual control system, whether this dual control system becomes more stable or it shows different kind of profiles. So we use this kind of contrast based on the Go trial and the stop trial, because both kind of trials are involved with proactive control. We define the commonality of activation pattern as per control, and we use the contrast between stop trial and Go trial to define reactive control. So we can use the single trial model and the representation similarity analysis to a searchlight analysis, so across the whole brain to see how this kind of patterns change from different developmental stage. So if we focus on reactive control, we didn't see much difference from the 10 years old to 12 years old, but we do see a significant increased neural stability of reactive control from 14 to 18 years old, including the anterior insular, inferior front gyrus, pre-SMA, and PCC, and basically the default mode, silence, and the frontal part systems. So we can also do a same analysis with the proactive control. So here we found like the widespread developmental differences from 14 to 18 years old. That's a dramatic improvement or increased neural stability during proactive control. So if we summarize our main findings from the reactive and proactive control, so we see there's an increased proactive control in the whole brain levels from 10 to 12 years old, from 14 to 18 years old. Whereas the reactive control is relatively stable during the early late childhood to early adolescence. So this profile tells us there's a long nonlinear developmental trajectory for dual control systems. So next, as I mentioned previously, proactive control is not a static process, but dynamically adjusted from trial to trial. So now we also know proactive reactive control, they are engaging similar brain systems. So we can basically use this knowledge to develop some metrics to estimate how much in Go trials they engage in dual control, right? So The way we did this is we first develop a template based on all the stop trials. Because in stop trial, the stop response, there's a strong inhibitor control. So we define this activation pattern using stop trials. We ask in every goal trials whether their activation pattern is similar to the average of stop trials. We hypothesize the more similar to the activation pattern to the stop trials, then the RT becomes longer. So this becomes a index, a neural index of inhibitor control, right? So we want to see whether this index has correlation with response time. This is what we found. So at every developmental stage, we see a highly similar pattern. This pattern, what does it mean? It's like a greater similarity to the stop signal, stop trial activations, the longer the RT it is, the slow response it is. So this is a trial-wise RT modulation and how the brain systems involved in modulated trial-to-trial behavior. And as you can tell, the main systems, including the salient and the frontal part systems, and also default mode. This is a developmental change, a difference between the two developmental stages. So we can also use this trial-wise modulation of the brain function to predict the trait-like behavior, for example, the SSRT. And here we use the elastic regression models, and we can show that both models provide the robust and significant prediction for each developmental phase. Here, different color represent different developmental stage data. There's a baseline is 10 years old, the follow-up is 12, and also for imaging data set. And the same for the response variability, the IRV. We can also look into whether this behavior prediction power changes across development. So as you can tell, so here, the y-axis is the accuracy, is a correlation between the predicted and observed behavior data, basically the power of the model, right? So you can see from the 10 years old to 22 years old, we observe the increased prediction power of behavior. And so what does it mean? So to me, I think it means like with kids development, so their brain activities are more efficient in modulate trial-to-trial behavior, which has a direct consequence to predict the trait-like behavior, like a behavior stability and inhibitor control functions. Because inhibitor control and the control function are widely implicated in different kinds of internalizing and externalizing disorders, we can further examine whether those kind of trial-wise brain modulation features can predict clinical symptoms. But because there are so many of the clinical measures across DSM and all kinds of questionnaires, so we use the canonical correlation analysis to look into canonical features can predict the clinical symptoms. So here in an ABCD study, we use the CBCL and the UPPS impulsivity scales, and we did find the significant correlation between the brain measure and clinical measures. Notably, so here in both brain measure and behavior measures, we look into a difference, developmental differences. So from 10 to 12 years old. And the same for imaging, where you look into difference between the different developmental phase. This one's 14 to 22 years old. And the features, so in both data sets, we found like the main features, including the anxiety, ADHD, and depression for the clinical measures. And on the brain side, there's anterior insular and the cingulate cortex and temporal gyrus. So this study tells us there's a high predictive power of the trial-wise behavior modulation can predict the clinical symptoms and also can predict the developmental change of clinical symptoms. So how the clinical symptoms evolve through developmental phases. And we also show there's a strong reproducibility across different data set and across different developmental stage. Okay, so in this first study, I demonstrated there's a nonlinear developmental profile for neural control systems, and we can build a behavior prediction model based on trial-based product control system. So in next study, I want to show you our recently published study where we demonstrate ADHD has a show a deficit in dynamic proactive product control systems. So here we use the same like the conditional stop sync task introduced previously, where we can better decompose reactive and product control components from the paradigms. So this is the data we collected in our own lab. We have 26 children with ADHD and 35 TD children. And kids on medication will go through a washout period, and we use the multi-band EPI for the data collection. So we use the same single trial and representation similarity analysis to look into a temporal stability. So basically whether the activation amplitude is stable across trials, and also the spatial similarity, where the spatial pattern is similar across trials. So our main findings first, the children with ADHD shows increased temporal variability. So from trial to trial, ADHD kids, the activation amplitude will vary more compared to TD children's, which matches our prediction. And we can see the main differences is in the frontal, parietal, and salient systems. Next, we also demonstrated there's a weak spatial stability. So from trial to trial, the activation pattern become less similar for kids with ADHD compared to TD children. This is the same dynamic product control analysis to look into which brain regions can track trial by trial RT modulation. This figure shows you can see two rows. In the top row is the TD, typical developing kids. In the bottom row is the children with ADHD. You can see it's a very dramatic difference between the two groups. For TD children, there's a significant brain activation patterns in the dorsal medial prefrontal cortex anterior insular inferior frontal gyrus. So those brain systems are constantly engaged, track the trial by trial RTs. And whereas children with ADHD is a much weaker patterns. So this is the contrast between TD and ADHD. We can look into whether there's a group differences on the ability to track the trial by trial RT fluctuation. You can see there's a significant differences, whereas TD children has a much stronger significant activation patterns in the dorsal medial prefrontal cortex and parietal regions, anterior insular, and the middle frontal gyrus compared to children with ADHD. And on the right side, this is just an example data point for one of the TD subject where we focus on the pre-SMA, which is here. And we look into how the single trial data is reflected the brain activation patterns. So again, here is the input control pattern. So the higher value means on these go trials, the activation pattern is similar to the stop trials. And lower value means less similar, dissimilar to the stop trials. Whereas when a similarity increase, see the RT is increased. So last, we can also demonstrate there's a pattern similarity with the ADHD, like neural patterns. So here is the figure for the right anterior insular. This is one of the regions we constantly find in our study. We show here as this pattern is more similar to the ADHD groups than there was the increased inattention symptoms. So in this study, I present some of our previous evidence demonstrated the children with ADHD shows reduced stability in both temporal and spatial features. So I want to contrast our study with previous work which people mostly focused on the average task activation but here we provide a dynamic view of the ADHD neural patterns, which can provide us more potentially clinical useful signature to understand how the behavior changes between two groups. So in last study, I want to introduce a previous finding. We use stimulant medication to look into how the methylphenidate impact the brain state dynamics and cognitive control in children with ADHD. So this is a collaboration with one of international scholar from Japan and Dr. Yoshifumi Mizuno. So he is from Fukui University. And so in this study, we don't have a task fMRI, so we only have a resting fMRI data. And but we do have scanner cognitive control task, which is a continuous performance task. And in this study, they recruit a group of children with ADHD, a group of TD children, everyone going through the MRI scanner for resting scans and complete the task out of a scanner. And for every kid, they have 2 sessions. It's a placebo-controlled double-blind clinical trials. In one session, kids take one single dose methylphenidate. In another session, a kid taking takes placebo. So the 2 sessions are counterbalanced and blended. No one knows which one is which. So in the end of the final sample, we have 27 children with ADHD going through the placebo and medication session. We have 49 TD kids. So here's the behavior. So behavior here is standardized T-score. This is omission error, commission error, RT, and RT standard deviation. So the higher score means the worst performance, okay? So compared to the placebo, you can see the methylphenidate can improve performance across different measures. We have also looked into some basic dynamic measures using the low frequency fluctuation. So basically measures both signal amplitude in a low frequency band. And our hypothesis is the methylphenidate will enhance the amplitude fluctuation of key brain systems the medication will be acted on. For example, the nuclear components, which is the reward motivation system, and also the cognitive control systems. Indeed, we show that compared to placebo conditions, methylphenidate does increase the ALF amplitude fluctuation in nuclear components, and as well as in the cognitive control systems, in the salience and default mode systems. So next, we want to understand how the methylphenidate modulates brain dynamics. So this is the resting state fMRI, so not task. So we cannot apply the same single trial model on that. So in this study, we used the previously developed a method from my former colleague, Dr. Jalita Hill and Dr. Shukran Rally. And they developed this Bayesian switching dynamic system. This is an unsupervised learning algorithm. Basically, they can automatically determine the latent brain states based on the activation and connectivity pattern of a time series, of fMRI time series. And in the previous studies, we have replicated and validated the paradigm and the algorithm using the simulation data and using the rodent optical fMRI data. And we have applied the method in the human connectome working memory data set to demonstrate the uncovered brain state can predict behaviors in a large data set. So here we apply this method in the resting fMRI data where we define 11 ROIs from salience, frontal parietal, and default mode networks. And on the right side, you can see this is basically the brain state evolution map. So each row represents one subject. I group them by the MPH methylphenidate group, placebo conditions, and the TD kids. And the y-axis is actually fMRI time point. So here we have about like 200 each time point. And each color represents the different distinct state identified by the algorithm. So here we have 5 different states. So we can look into the property, temporal property of the state, like how often the state occurs during time, and how long the state lasts. So what we found, our main finding is compared to the TD groups, ADHD in placebo has increased the occupancy rate on certain brain state, for example, state S2, and whereas the methylphenidate can reduce this abnormal increased state occupancy rate, bringing to the level very similar to TD controls. We can also look into the connectivity profile, which defines the latent brain state of this S2. And compared to a placebo and the TD groups, we see there's a enhanced connectivity within the default mode systems in the placebo group compared to TD. When we compare the methylphenidate with placebo, we see it can cooling down the connectivity and reduce the connectivity, not only within the default mode systems, but also widely across the salient network and the frontoparental systems. In the end, so we also show that the methylphenidate effect on the functional connectivity between the anterior insular and posterior cingulate cortex can predict the medication effect on the IRV. So this is out of a scanner task. So we measure how the medication affects the standard deviation of performance, remember the medication, and reduce IRV. And here we see the medication effect on the dynamic connectivity directly associated to the medication effect on response variability. So in summary, so I presented 3 ongoing and recently finished study. So I talked about like the neurodevelopmental of the neural control systems. So we see that distinct developmental profiles, behavior and neural stability continues to stabilize and decrease from late childhood to early adolescence, whereas the neural control systems exhibit dissociable nonlinear patterns. Importantly, we demonstrate a dynamic product control. The brain mechanism can predict trait-like behavior as well as clinical symptoms. And in second study, I showed written publications that children with ADHD, not only the distinct neural features of instability was demonstrated with the increased temporal variability and reduced spatial consistencies. So in last study, I presented some methylphenidate results showing how the methylphenidate improves the upper brain dynamics in children with ADHD. And last, I want to thank my collaborators, especially Dr. Riley, Dr. Gao, Dr. Hill, and Dr. Mizuno contributed a lot to the studies. I want to thank all the funding support of my research and also the public data is important for all of us. Thanks for your attention.
That's such a wonderful talk. Now we are open for questions. For in-person attendees, please just raise your hand if you have a question.
Go ahead.
Thanks so much for a wonderful talk. I was hoping you could comment a little bit on the role of the DMN in inhibitory control. I noticed it came up semi-regularly in your findings, and I'm just wondering about the role of that network.
Right, that's a great question. So the DMN roles in inhibitory control. So in typical task fMRI study, people try to focus on the highly active brain systems. So in most of the studies, as you can see in the meta-analysis I present, because people focus on the activation, so usually the findings mostly focus on the salience and the frontoparietal system, such as anterior insular and pre-SMA. Whereas DMNs, we know that DMN plays a lot of roles in cognitive control, especially based on the connectivity analysis. People have shown that the connectivity between DMN and also the salience network and frontoparietal are important, modulated by the task conditions. And there's also some study shows, like from David Sharp group, that the anatomical disruption of the connectivity between the DMN and anterior insular kind of affects the SSRT. So there's a lot of research, but not in the activation profiles. But in terms of connectivity, there's a lot of research into how the connectivity between the DMN and the sense networks modulate the ability to load or switch different cognitive states.
Thank you. We have a question from Zoom. Is from Nicola Sharp. Apologize if I mispronounce. Is there indication from the data as to how feminine background and home environment may impact development of proactive control? Were these factors taken into account in the predictive models?
That's a very good question. So no, we haven't looked into all the potential other factors like environmental factors and how it's going to impact the developmental proactive controls. But I think for the ABCD study, there's a lot of data there, so we can explore to those conditions. Yeah. Yes.
I'm just curious if you looked into gender differences. I mean, I saw you had both.
Right. I think that's such an important question, especially for me to study ADHD. You know, the gender is a big factor to consider. And in the current analysis, we haven't deeply looked into the gender differences, but in our previous analysis, we do show that the gender, the girls and boys, they have some distinctive profiles in terms of connectivity when we try to predict the clinical symptoms or determine whether it's ADHD or not, but not in the current study. But we are looking in that. Yeah. Thanks. Yeah.
Another question from Zoom, from Nicholas Wheeler. ADHD seems to be context dependent. Kids can be deeply focused on activities they are interested in, but struggle to focus themselves to things that they are not intrinsically interested in. How do you see this individual context? What activities are more or less compelling play into the story of cognitive control system?
Yeah, I think that's a very important question. As we know, the question very well elaborate. So kids could be very engaged in some kind of activity, but not in others. So I think there's a multiple layer of the reasons. One is that ADHD is such a heterogeneous disorders, and there's a subgroup of kids maybe represented better by different distinct neural deficit compared to others. And at least for us, we know there's two major series in the field. One is the implicit control, which I focused in my talk. Another big series is reward and motivation, right? So a lot of people think, so implicit control deficit may be a secondary to the motivation deficit. So some kids may just be not interested doing all kinds of the effortful tasks. So I think that's an important aspect to consider. And in terms of the questions, I don't have a clear answer to that, but we show that in the medication study, the methamphetamine does enhance the spontaneous body activity in the reward systems and in current control systems. I think with the larger scale data set, one opportunity is so we can look into heterogeneous profiles and say what kind of kids are susceptible to specific deficits compared to others.
Thank you so much.
Yeah, you were talking about the temporospatial besides the stimulant medications, what other kind of interventions have you devised to intervene to help those individuals regulate better at those moments?
You're asking the intervention method? No, we're not in that stage yet. So we're just doing the imaging study, looking into the reliable biomarkers, whether we can characterize at least the subgroup of kids and show the instability of behavior and neurons. I think it provides us, so the importance of this study is it provides us some novel biomarkers, which we can develop a future intervention method to target specifically the dynamic deficits of the brain system rather than focus on the static measures. Yeah, thanks. A great question.
We have one more question from Zoom from the same Nicholas Wheeler. When you say you observe brain state dependent hyperconnectivity in default mode network, could you clarify what brain states you are referring to? What brain states specifically affect the default mode connectivity?
Right. So just to clarify, so this is the last study I was talking about. The brain state is defined by the unique pattern of activity and connectivity among a group of well-defined brain regions. For example, Silence, frontoparietal, and default mode networks. So brain state is represent a unique combination of the features. For example, some state maybe show the tight connectivity between the default mode and sense network. Other state shows the less connectivity of that. So when I say the hyperconnectivity, it's when I compare ADHD with TD groups, which will show the connectivity is increased compared to the normal kids. Yeah.
Thanks for such a great talk. That was awesome. Quick question. How do you interpret, so you showed that those nice developmental results that showed this kind of increase in stability, in particular, during adolescence. And the increased stability was kind of seen across a huge chunk of cortex, not necessarily kind of specified or kind of region, not necessarily within kind of specific networks you might have imagined. How do you interpret such widespread changes in stability across cortex?
So yeah, that's a tough question. So I want to clarify that there's a different stage comparison. We're looking into one ABCD study from 10 to 12 years old. We didn't see much change of that. And the biggest change we see is imaging study from 14 to 22 years old. So we did see the wide spread of the difference between the two developmental stages. But I think that's mostly driven by the threshold that we pick. So if we increase threshold, we'll see more focus. But in general, I think there's from adolescence to the young adulthood, there's a substantial increase stability in the behavior side and also in your system side. Yeah, but I agree with you. So I also have some same doubt as you have. What happens to the whole brain? So let's go. Yeah, thanks. Yeah.
Thanks.
Okay.
Readings showed state-specific default mode involvement in inhibitory control. Do you interpret it as a failure of suppression, failure in switching to Silence network, or difficulty disengaging the default network?
Well, that's a good question. So the quick answer is I don't know what this mean at this stage, but based on our hypothesis, like default mode and sense networks, they are involved in like the switching different states based on the task need. But as we showed in our data in the resting state fMRI, so that's not the time point where people actually engage a task. So we are just showing some intrinsic brain dynamics. So I don't know how it's to directly link to the task state brain system function, but I suspect it's aligned with what the audience has proposed. So perhaps it's linked to the difficulty to switching the system to the proper task. Yeah, thanks. Yes.
In the medication study that you talked about toward the end, I don't know that that you did this, but I'm curious if the effect of the medication is only while the child is on the medication, or if there was any kind of pre-post look to see if there was change in the operation of the brain after medication, but not on it.
Oh, so if I understand your question accurately, so this is a single dose medication study. So they didn't, also those kids are the medication naive kids. So they didn't have medication before. They just took one single dose medication and wait for like 4 to 6 hours and go into scanner for a task. So they were not looking into the long-term effect. Yeah, good question. Yeah.
Okay, is it this on, right? I wanted to just reinforce how important it is, the work that you're doing, and to look at it from this dynamical approach, also to give a tangible neural basis to ADHD, because clearly it's such a prevalent condition and it's thought of historically. It's we haven't had a tangible way of thinking about how do you understand what's happening in the brain that you can link to symptoms, behavior, and treatment decisions. So it's incredibly important. Thinking of among the many opportunities you have in your future research, you touched on heterogeneity, and that, in my mind, is another really important aspect. So I wanted to connect it to some findings we've recently had. So in one of your slides, you showed the link between different types of symptoms and the brain regions in the stop signal task. And it was interesting to see that when you look at conditions like depression, you're bringing in other aspects of cognitive control, dorsolateral prefrontal cortex, anterior cingulate cortex. And I highlight those because when we look at adults, we see those regions and parietal disrupted for people with depression who have similar kinds of cognitive control problems as ADHD and who respond well to guanfacine, which is also, at least in extended form, a treatment used in ADHD.
Right.
So my question is, would you think among all the future opportunities, given the size of your sample, you could stratify the sample based on either different profiles of activation and connectivity when you scan, and also look at whether those different stratified profiles might link to different types of symptoms or maybe associated symptoms? You could have ADHD with relatively more or less depression.
Yeah, I think definitely, like that's the one of the goal of our research direction. We want to apply our findings in the clinical practice and to help patients and families. So I think this requires a larger scale of studies, such as ABCD studies, right? And we can have large enough samples to identify the subgroup and heterogeneous groups, can develop more characteristics associated to the specific groups. So the idea is to do that, move into that direction. And I know I mostly study ADHD as like just want to bring everyone's attention. ADHD, where now it's a very, very, very common disorders, like based on the NIMH surveys, this is like about 10% of kids, based on parent report, they have been diagnosed with ADHD. And if you look into a specific age group, like 9 to 10 is a boy, maybe like the rate could be 15%, which is kind of scary. If you imagine in a classroom in elementary school, like the 20 kids, you know, the 2 or 3 kids has ADHD, and the classroom is very hard for teacher to control with, right? So, but again, the control deficit is the transdiagnostic. It applies to all kinds of disorders, including depression and anxiety. I think how to develop this transdiagnostic features, we can, based on the non-invasive imaging method, and develop corresponding intervention approach, as definitely important. And so that's something we hope to work on. Thanks. Yeah.
Okay, thanks again for coming despite the rain. And thanks to our online attendee. Let's thank our speakers one more time for this wonderful talk.